Hello, everybody. I can see everyone's filtering into the meeting now. We're not going to start just yet. We'll let everyone get in, but welcome and thank you ever so much for joining us this evening. We had about 50 people in the waiting room, I think. I can see 41 people are in the meeting now, so we'll just give it a minute more so nobody misses everything. Gosh, we're over 50 now. That's brilliant. All these Dryopteris enthusiasts joining us. Right, that seems to have settled down now. Hello, everybody, and thank you ever so much for joining us for another of our webinar series that we've been running. This webinar series has been made possible thanks to our Northern Ireland Botanical Skills Project, which is a project that is looking to support botanists across the skills spectrum with training, events, and online webinars like this. And thank you to our funder, DARA, for making the entire project and this series of webinars possible. This is the fourth webinar in the series. I think as you registered and received emails, you should have received a link to the rest of the webinar series. But if not, have a look on the BSBI events page and you should be able to see other webinars. They're going on until beginning of February next year, and there are other exciting talks to look forward to. Next Tuesday, we've got Robert Northridge talking about equisetum, and I know that next year we've got some talks on getting started with grasses, sedges, and rushes for beginners, but we've also got talks on rubus, brambles in Northern Ireland as well, for those for those braver than me. There will be a Q&A session at the end of this webinar. So you'll notice that there's a chat at the bottom and there's a Q&A at the bottom. As we are going, feel free to put questions in the chat as we go along. I will only serve up those questions at the end, but it's nice to have a few to get started with once we reach the Q&A session. And I think that's kind of all the housekeeping we need to do. Luckily, we don't have to do, fire alarms and fire exits and so on when we're doing online webinars. So I'm going to introduce you all to Alison Evans. So Alison is a retired GP with a lifelong interest in botany, now working as an associate tutor at Edge Hill University. She has a particular interest in the Dryopteris affinis complex and is now partway through a PhD on the morphometrics and taxonomy of this complex, which still turns up surprises on a regular basis. And so I am delighted to hand over to Alison.
Great, thank you very much, James. Can everyone hear me? I hope. Yes, good. I'll share my screen, I hope. That one. That one. Okay. Yes, I don't need that. Great. Thank you ever so much for asking me to do this talk. I think I should say right at the beginning that I've spent a total of 1 week in the north of Ireland looking at ferns. And this was on the British Pteridological Society trip there in 2017. So I've really had to draw very heavily on the BSBI distribution maps. And also, has James put his hand up? No, okay. In order to know exactly what there is in Northern Ireland, but most of my photos are from elsewhere. So I would ask you, if some of the things that I say don't fit with your Northern Irish experience, please just put a comment in the chat or a question at the end. So my plan, why can I not move on? There we are. I'll start by concentrating on the ones that are found in Northern Ireland, the buckler ferns and then the male ferns, and then we'll have a quick look at other Dryopteris, some of them not yet recorded in Northern Ireland, but maybe they will be. So starting with one that I expect that most of you know, that you're familiar with, a really common fern, often the dominant fern in more acidic woodlands, but it isn't picky, it turns up all over the place. The distinguishing features here are the dark stripe down the middle of the scale. And that tends to distinguish it from the other buckler ferns. It grows in a shuttlecock. Being a Dryopteris, it has the kidney-shaped indusia, but they do fall off, they shrivel and fall off quite early in the season. And so it can be difficult to tell what shape they were. The other thing that's easily recognizable is the way that the pinnule edges turn down. So it looks like there's little roofs. It's often epiphytic, one of our few epiphytic ferns that we have here. I think you can see that turning down appearance better on that photograph. And as you can see from the distribution map, it's ubiquitous, really. An interesting thing about this fern is it's a tetraploid, and you probably know that ferns tend to hybridize, and then they can overcome their hybrid infertility by doubling up their chromosomes. So back in 1983, Mary Gibby did some work with hybridization and worked out that probably this fern is derived from Dryopteris expansa and Dryopteris azorica, neither of which occur in the north of Ireland, I don't think. Now, recently, that's been confirmed by some DNA sequencing that's been going on in a project that's led by Michael Kessler in Zurich. Moving on, the narrow buckler fern, really quite common in wet woodlands. It's narrower than the broad buckler fern, and it has these rather flat pinnules. The scales are pale. It still has the kidney-shaped indusia. And this is the fern that you can recognize from the train window or the bus, because it doesn't grow in those neat shuttlecocks. It tends to have a little clump, and then it wanders off. So the fronds point in different directions. And the other thing about it is the pinnae tend to be rotated to the horizontal, so it looks more like a step ladder. Now, if you're not sure which one you have, then you might need to get your hands dirty, go right down to the bottom of the fronds and look what the croziers are doing. So we have these dark scales over the croziers that all point into the middle in the broad buckler fern. But Dryopteris carthusiana has these more greenish croziers with pale scales, and often there's sort of 4 of them at right angles to each other, but they aren't arranged in the neat crozier. And looking at the distribution, you can see that Dryopteris carthusiana is quite common as well. It's always worth looking for in wet woodlands. And one thing about Dryopteris is it's really good at hybridizing. So where these grow together, it's quite common to find their hybrid. which is named after Mr. de Vever, and we anglicise it to Dueveri. Just an aside, I took this picture from Teredo Portal. This is an American site. I think it's really useful. It's all of the digitized scans from several of the US herbaria and also fossils. So it is quite a useful site. But to look at the real plant, James Merriweather says if you can't decide whether it's narrow or broad, then you've probably got the hybrid. The scales tend to be a bit paler. It's almost a shuttlecock. It's a bit more upright. And if you look down at the croziers, they can't really decide what they're doing. A bit palish, some a bit dark, and trying to do a bit of a crozier there. Excuse me. Now, if we look at the distributions, these two ferns grow together a lot in the north of Ireland, and possibly it's under-recorded. If you look at the distribution in the UK, in Scotland, England, and Wales, it seems to occur in clusters. But I think I can probably name the pteridologists that have made those records in those clusters, at least some of them. So I would encourage you to have a good look when you're out in these wet woodlands and see if you can spot the hybrid. The ideal time is to go when the spores should be ripe, because of course you'll find abortive spores in the hybrid. And then on to the fern that I really envy you, because it's quite common in the north of Ireland, and I have to travel quite a long distance to see it at all. The hay-scented buckler fern. I've never smelt the hay scent. It's supposed to be on herbarium specimens, but I've never been able to pick it up. So it has pale scales, and the sort of distinguishing immediate feature of it is that the pinnules are nearly always crimped upwards. And if we look at the distribution, it's quite a maritime thing. It does grow in some lakeland woods in Cumbria. In my experience, if you find this, you can often find hymenophyllums growing nearby. So it's always worth looking. If you find one, is the other there? It tends to be on very humus-rich and steep wooded slopes. Can be quite tricky to get to. Okay, so deep breath for male ferns. People often say to me, why are you studying male ferns? They're not very pretty and they're boring. Well, I don't agree. The picture on the left is a whole collection of different diploid scaly male ferns catching the light at the bottom of the waterfall. And I think you can just spot Roger Golding, who's been just such a help in field work over the last few years. And then the picture on the right, Dryopteris palliaceus lobata, is quite architectural, I think. I have some in the garden. I think they're really a very good garden plant. So to start at the beginning, maybe, the common male fern This is a twice divided fern. We call it bipinnate because the pinnae are just once divided into pinnules. It doesn't have a dark spot where the pinna joins the rachis. And it is variably scaly. Some of them can be really, really scaly. You wonder if you're looking at a scaly male fern. Generally, the texture, though, is much thinner and more papery. This is really variable. And again, this is because it's a tetraploid derived from two different diploids, Dryopteris oreades and Dryopteris caucasica. Again, neither of which occur very much in the north of Ireland, as far as we know. Those two genomes are not entirely different, and so there can be crossing over between them. And I think that's probably why this fern is so variable. It doesn't really make nice shuttlecocks. It tends to grow in clumps. If you look at the indusia, the coverings over the sporangia, when they start out, they often look like the sort of soup plate that has a rim around the edge. So the edge is sort of spread out along the lamina, and they're often rather crowded together. They turn grayer as the spores mature, and then after the spores have been shed, the indusium tends to go brown and shrivel, and they drop off quite early. It's a very common fern, often in neutral to more basic woodlands, but it grows everywhere. It grows on walls. As you can see, it's pretty ubiquitous. And I've compared this with the distribution of the aggregate Dryopteris affinis as recorded on the BSBI website. And it's good that it's recorded so much, but wouldn't it be good if it was actually recorded as the individual species? As they occur in many places together, I'm afraid they do hybridize. And for every different affinis taxon, there is probably a hybrid with Filix mass, but I won't be going into details of those this evening. So one of the difficulties with the affinis is, what do we call them? And in the good old days, it was fairly easy. There was one species and 3 subspecies. This didn't quite satisfy people who found lots of different forms of scaly male fern. And Clive Jermy, in the guide he wrote in 1991, and then Anthony Pigott suggested that these different types should be called morphotypes until the work was done to clarify their genetic relationships. Now, morphotype is an illegitimate botanical term, and Christopher Fraser Jenkins wasn't happy with that. He published a paper in the Fern Gazette raising those subspecies to species. And the different varieties that he recognized, he made into subspecies. So this didn't really solve the problem of the varieties that didn't fit. And Ken Truran, who looked at a lot of Dryopteris affinis, compiled this guide. Sadly, he died in 2010, and this guide was published posthumously. So he included all Fraser-Jenkins subspecies and the morphotypes in this guide. It's out of print, but you can download a copy from the BPS website. That's eBPS. Should have said that, sorry. Okay, but then Peter Sell decided that as these plants probably had different lineages, that they should be separate species. And so we have all the previous subspecies now made into species. The following year, though, Clive Stace was persuaded by Fraser-Jenkins to put them all back into one species again. And this has meant that quite a few of the things that we refer to as subspecies have now just become varieties, and so tend not to be recorded any longer. There is a difficulty with this in that certainly the pteridologists in continental Europe still stick to the 2007 scheme, adding in taxa that have been published since. So, coming on to the in the strict sense, Dryopteris affinis, which we could also call affinis subspecies affinis var affinis. It's a very architectural plant. It usually forms a lovely shuttlecock. It's quite robust normally. And if you look at the distribution map on the BSBI website, it seems to grow in clusters. But again, I think I know who's responsible for most of those clusters. So I think it is a matter of where knowledgeable people are recording, really. And how do we know that it's a scaly male fern and not a common male fern? This dark spot where the pinna midrib or the costa joins the rachis is a good guide. It's not absolutely foolproof, but it's a pretty good guide. And one thing that is often helpful is to look at the lowest pinna and that first basiscopic pinnule here. And in affinis, it's usually up to a half joined on to the costa. It's a very glossy and leathery frond with impressed veins. Now, Tony Church came up with the idea, if you hold a pinna with the top side toward you, hold it up to the light and look at the veins. The veins of affinis are usually really clear. Tends not to be very toothy, but it can be in some clones. It has tall, very thick indusia that are tucked under at the edges. And as the spores mature, these often rise slightly, and there may be some small radial splits, but the indusia tend to stay on the plant until, well, the following year. Often you can find a brown frond and it'll still have the indusia there. So moving on, Borah's scaly male fern often grows in big untidy clumps. But the young plants may form a reasonable shuttlecock. It doesn't tend to be as upright or as, what shall I say, rigid as the affinis. It's very common. And it's very, very variable. And the reason for this is that it has 3 different genomes. So it has the Oreades and the Caucasica that Filix mas does. And it also has the affinis ancestor, which gives it these, the dark spot. So if you look at this lowest first basioscopic pinule, it's not so adnate. There's usually just a broad stalk. And the picture of the mid-frond there shows a feature that a lot of people look for as an indication of borari, and that's the truncate pinnules looking as though they've been cut off with scissors. Now, we maybe refer to that as Newman's borari because it's similar to the one that Newman described. But this appearance is not diagnostic in itself. And I have seen other taxa that are also quite truncate. So it's part of the picture, but it's not in itself diagnostic. And if we look at the sori, they tend to be like a sort of pudding bowl with the edges just going down to the lamina, not tucked under and not particularly spreading. And after the spores are shed, the indusia tend to shrivel up. If we look at the veins again, they tend to stop short of the margin of the pinnule, and they have areas that are not very clear. And I've put up a couple of types of borari here. The one that has the truncate apex and the cat's ears, these rather sharp teeth in the corners that I think Clive Jermy described. But you can also get these rounded apices, again often with the sharp teeth. And coming on to the third species, I'll call it, this is Dryopteris cambrensis. Tends to be upright and a bit untidy, fairly common in the north of Ireland, very common in Scotland, probably more so than in Wales, so odd that it's called the Welsh scaly male fern. Looking at that lowest pinnule again, it's stalked usually, and the dark spot can run up to the 3rd or sometimes 4th pinnule. It's really variable, and there is a question as to whether it should actually be separated out. In all of them, the pinnae tend to rotate a bit, and often the pinnules are turned upwards in relation to the rachis. There are some forms that are really quite flat. The auricle tends to overlap the rachis, And some forms are really crimped. And by that we mean that the pinule often turns under in the middle part and then sort of opens out into a rather spoon shape. When you look at the veins, they're not quite as distinct as in affinis, but they do go almost to the edge. Some clones are quite toothy and others aren't. The Indusia, they do look as though they're tucked under. And after maturity, they're not quite sure what they do. Some of them rise like affinis, some of them go to cones like boreri, and some of them do something in between. Now, this picture was taken in the south of Tipperary, actually, just by the roadside. And this is a mature clump of Dryopteris cambrensis that demonstrates, I think, it's quite an untidy thing. The fronds, the rachis tends to be quite twisty rather than being straight. And I think you can see that the pinnae there are turning upwards. So just to compare those three with the venation, in affinis, there's clear veins that go virtually to the edge of the pinule, much less clear in boreri and a bit intermediate in cambrensis. And thank you very much to Tony Church for those photographs. Now then, when it comes to deciding which taxon you have, it seems that the average length of the stomata is quite a good predictor. So the diploids, it tends to be 45 microns, or at least in the mid-40s, sometimes lower. The triploids tend to be around the 50-51 mark. And the hybrids, which are either tetraploid or pentaploid, tetraploid 56 to 58, and then up into the 60s often for the pentaploid hybrids. Measuring stomata is quite easy if you have a microscope. Once you've learned how to strip off the epidermis and then put it in glycerin on a slide, cover slip, let the bubbles all disperse, often a day or two. And then if you have a graticule in your eyepiece, you can measure how long the stomata are. I have to say that in the project I'm doing, I've been greatly helped by Libor Eckert in the Czech Republic, who has done flow cytometry. So I've had the ploidy level confirmed on virtually all of the samples I've collected. So going further afield, I was slightly surprised to find that Dryopteris expansa hasn't been recorded in the north of Ireland. Not sure why that should be. There's also Dryopteris submontana, and that's very localized around the limestone pavements of the north of England, and one or two stations in Wales and in Derbyshire where it grows on railway cuttings. So Dryopteris expansa, the northern buckler fern. This was taken on a very wet and misty day on Ingleborough. I think you can see on here that this first pinule is really long. It's almost as long as the rest of the pinna. So that's a character of expansor. It's usually a bit more dissected than dilatata, and it has paler scales, but they can be really difficult to tell apart. And this picture was taken near Corriffy in Scotland. If sometimes the scales are darker than you think they should be, it does have smaller spores, and they're rather paler than Dryopteris dilatata. It is a lovely thing, and I'm sure you've been looking out for it because it would be great to find it in the north of Ireland. The other one, Dryopteris submontana, it's a very glandular plant, and it really is a limestone pavement specialist. And the way to tell this is how glandular it is. It gives a grey appearance to the whole fern, really. And I think you can see even along the costa there, there's, no, sorry, the rachis, it's really glandular. And a few more male ferns then to look out for. I have looked very hard for Dryopteris oreades in the south of Ireland and not found it. But I think probably subspecies Paleaceolobata or Dryopteris paleaceolobata you may well have in the north of Ireland. And subspecies Kerriensis is not just in Kerry, it's quite widespread, but we'll come on to that. So the mountain male fern, when I learnt my ferns, I thought of this as typically a plump forming, rather small, upright fern of high screes. The sori, there's rarely more than 6 on a pinule, and they're right on the inner half of the pinule. This photo from Jeremy Roberts, I think you can perhaps see the little white drumstick glands around the edge of a really tucked under indusium. So the indusium looks a bit like a mop cap. I had to change my idea about Dryopteris oreades when I saw it growing to the same height and looking very similar to the common male fern in Borrowdale in the Lake District. And the picture on the left is that same day on Ingleborough, but I think it shows how in the sori near the middle of each pinna, how they form, it really looks quite stripy. And this was quite a big floppy plant as well. Now, Roger Golding's been doing some work on this because Dryopteris oreades, it's one of the parental species of Dryopteris filix mass, where they grow together. Yes, they hybridize, and that's Dryopteris crasmantonii. There are a few records of that. I haven't put them, sorry, I haven't done a distribution of that. But recently, Roger sent off 20-odd samples for flow cytometry, and 4 of them came back as the triploid hybrid. So the hybrid is out there. Okay, I talked a bit about Dryopteris palliaceolobata, or affinis, subspecies affinis var palliaceolobata. I think it's a really handsome fern. Tends to be very straight and regular, with regular crimping. Unlike affinis, it has a stalked first lowest basiscopic pinule. The indusia tend to split and go up to form these butterfly wings. As I said, the crimping looks really regular, and the veins stop short of the margin, mostly has obtuse teeth. I don't know if it's becoming more common, or people are just recognizing it more, but it is worth looking out for. It's much more regular than cambrensis, but otherwise it can look a bit like cambrensis. And then another lovely thing, the Irish male ferns, so Dryopteris cariensis. The pinnae tend to go out at right angles. The pinnules are really congested. It often has these dark ginger scales. So to look in a bit more detail, statistically, it has the smallest, lowest frond width. So in the morphometric study I'm doing, I measure the mid frond width and compare that to the lower frond width. This one is statistically different from the other diploid taxa. It's really tapered to the base. And the indusia often do either butterfly wings or they lift. Around the edge of the pinnules, they have these, it's more toothy. And some of the veins are really rather indistinct, so it's unlike affinis in that respect. And if we look at the distributions of them, Roger Golding found cariensis in Cumbria, and there are quite extensive colonies there. So I think we do need to be looking for it in more places. We've also found one in Somerset and one up in Scotland. When I look at this distribution of Dryopteris palliaceolobata, I'm thinking, oh, well, that's Tony Church and that's Roger Golding, and this is probably Matt Stridley. I think we all need to go out and look for it a bit more. I'm also very conscious that we have some records in Galway that haven't appeared on the database yet. So that's something we need to think about. Okay, so 3 other things, 2 that I think will turn up in the north of Ireland, and then the last one's just a bit of interesting thing. So Dryopteris lacunosa was described in 2011 by Stefan Jessen, and the paper's in German. Fraser Jenkins has dismissed it as a form of boreri, but Stefan Jessen has grown it in an experimental garden for 25 years. It has a different phenology to Dryopteris boreri. It has some different chemistry, and it has different morphology. So I would be inclined to call this a separate species. The features, these double teeth on the auricles are quite common, but not on everyone. The pinna stalk can be up to 5 millimeters, and that's what gives it this really spaced out look when you see the frond. The gaps between the teeth are rather funnel shaped, and it's described in the paper as fjord shaped. The indusia tend to do much what Dryopteris boreri does. And then the hybrid between Dryopteris affinis, the diploid, and Dryopteris filix mass is incredibly variable. To give you just one picture, it can't capture the breadth of the variation of this. Again, I think this distribution map doesn't do justice to how common it is. It really is intermediate between the parents. It's more robust than filix mass. It's a bit more disorganized, not quite as leathery, and the dark spot tends to be rather less intense. Again, look for the spores. If the spores are abortive, then you probably have the hybrid. And just to look at in a bit more detail. So it's moderately scaly. The veins tend not to reach the end. It's quite filix mass-like in several aspects. And then thank you to Fred Rumsey, who has been over to Ireland not so long ago to look at Dryopteris remota. There's a very old record for this, and it's now been refound, I believe, in County Clare. Whether it originally escaped from a Victorian fern lover's garden, I don't know, but it is a really lovely thing. And I just thought I'd leave you with that as a, looking for ferns is exciting. You never know what you're going to find. Okay, that's it.
Thank you ever so much, Alison. That's wonderful. My first question, and I know we've got some questions probably being dropped in the Q&A.
I better stop sharing, haven't I?
Oh, yes,
I think.
yeah.
Good.
Excellent. So that text, that is the sum taxa within the Dryopteris affinis complex. Is that right?
No. There are at least 8 different diploids.
Oh, sorry, the name of the book that you were recommending at the beginning, was that sum taxa within the Dryopteris affinis complex? Is that what it's called?
Yes.
Yes.
The guide, yes.
Yes. Yeah.
And we found more things since then. 1 or 2 are published. In fact, the other interesting Irish one is Fraser Jenkins published subspecies Atropes, which has very black scales, but he's only just published that. I think it was 2022. New things appearing all the time. what I didn't say was with the Dryopteris lacunosa, just this year, the German group published its hybrid with Filix mass, which is called cross urbachensis. And looking back at our collections, we found a pentaploid in County Meath that really fits the description. So we're going to have to publish that, I think.
Right, right,
Sorry,
okay. So
yeah.
at the moment, there's no one single place to get an overview of all of the tax that you talked through, but that guide that you mentioned is a good starting point.
It's a good start. Yes, it is. Yes. Yeah. Um, and well, the next Fern Gazette, there's a review of recent literature.
So that could be really useful.
So,
Right,
yeah.
brilliant. So opening the Q&A, Paul Smith asks, do stomatal lengths work with dried specimens or are the lengths different?
It's almost impossible to strip the epidermis off a dried specimen. So I put them in water for about at least 5 hours. Take a dried pinule, put it in water, rehydrate it, and then it seems that the lengths are pretty much the same. There's a little project for someone to, look at 50 or so dry, fresh and dried. I could do that, I suppose.
Yeah, yeah, that could be very
Yeah.
interesting.
There is some shrinkage with drying generally, and I'm not sure it's been measured, but all the people who do look at stomatal lengths rehydrate their dried specimens before they do the measuring.
Brilliant. Thank you very much. I hope that answers your question, Paul. Um, Robert Potvlige asks, many thanks for the talk. Could all this variation indicate the existence of a number of cryptic species? Do these variations persist in cultivation?
Yeah, good question. Yes, probably there are quite a few cryptic species. A recent, well, in fact, it was the Urbachensis paper. They also reported limited genetic work, I have to say, but they seem to find two different lineages of Dryopteris affinis, with a different genetic pattern, but they were only looking at one gene. And the project that's going on at the moment uses target enrichment probes for flagellate plants. And these are very sort of clever DNA things that amplify the single copy or low copy nuclear genes. And then these can be sequenced. And that's what's going on at the moment. And at the start of that project, that's how they confirmed the Dryopteris dilatata parentage. But yes, in affinis, because they hybridize and the hybrids often have a bit of residual fertility, we are getting quite a lot of odd forms turning up.
That is fascinating. Thank you. And that's just made me wonder, and forgive me if I don't phrase it correctly because we're getting out of my comfort zone here, but is it possible that because some of the species you mentioned were of hybrid origin, is it possible that these have independently arisen more than once in different locations?
It certainly is. Yeah. Yes. and probably cambrensis is arising all around us because Oreades and affinis grow together. And Ken Truran named something called proto-cambrensis, which he thought was that hybrid. There's been interesting work done in the Czech Republic by Libor Eckert and his team. It is interesting. The apomixis, the sort of production of unreduced spores, isn't always what happens. And in the hybrid between Boreri and Filix mass, so that's cross critica, that can produce pentaploid spores and spores with between 2 and 3 sets of chromosomes, and they germinate, some of them. So it is, I mean, genetically, it's amazing. I think it's you know, a fascinating field of study.
And it really pushes.
Sorry, there was part of the question I didn't answer was, do these things stay true in cultivation? And the answer is not always. So it is difficult to unpick what is environmental and what is genetic. And that's why Stefan Jessen's work, he's had these plants in his garden for getting on for 30 years. And so he can see what happens. I'm hoping to establish a collection. Anthony Pigott has a collection. So, you know, the work still needs to be done.
Oh, that's fascinating. Thank you. Somebody has said that they've looked at the indusia of a male fern under a microscope, and it looked like they were covered in butterscotch and yellow banded worms, and they were all dead. Any idea what that
Okay.
could be?
Well, I'm just wondering whether some of the sporangia were still there, because you know there's an annulus on each sporangium that looks segmented. I'm just wondering whether it was that. But it's amazing what you see looking down a microscope. All sorts of things can pop up.
Absolutely.
Butterscotch. Not sure about that.
Heather asked during the talk, what does basiscopic mean? And I had a look at the Teratological Society website where it says pointing towards the base, but would you?
Absolutely, yes. As opposed to acroscopic, which is pointing upwards towards the apex. So acroscopic and basiscopic. Yes.
So it's not about being near the base, it's about pointing towards it, it's about the
Pointing that
angle.
way. Yes,
Yeah.
pointing downwards. Yeah.
Excellent.
Yes.
Thank you.
Sorry.
No. It's fascinating, isn't it? When you look into different groups, the kind of different language that pops up that you can be unfamiliar with from elsewhere. I think that's all of the questions in the Q&A. If anybody has anything, please put something in immediately if you want to ask it. If not, is there anything else that you'd like to, I don't know, promote to us, Alison? Is there anything that you think, if anyone's interested in ferns, they should go and do as a result of seeing this?
Well, I think there's there's an all Irish fern group has been established, I think. I'm not quite sure who is leading that now. I think check the BPS website, and I should have put that on. It's ebps.org.uk.
I'll put that
Maybe
in
we should
when.
put that in the chat.
Well, I'll put it when we put the video on YouTube. I'll put that as a useful link in the video description for
Yes,
people to explore. And
yes.
I'll put the link to the page with the some taxa within the Dryopteris affinis complex because I found it on the
Thank you.
website as well,
Yeah,
so people can
thank you.
find
Yeah.
that. Excellent. Um, Heather says she's not sure if it's off topic, but do the BPS and BSBI swap fern records? I'm not sure off the top of my head. I don't know if you know, Alison.
Um, we should all submit them to the BSBI, actually. So, yes, Fred Rumsey was our recording officer until recently, but it really is up to all of us to make sure that our records get to the BSBI, because that's, that is the place where the records are kept. There are a lot of records actually in our bulletins, the BPS bulletins, and they should be on the website, as on the BSBI website. Mm.
Excellent, thank you. And someone said the American website you mentioned, what was it?
Oh, yes.
Photo?
Teredo Portal.
Peridot Portal. Excellent. Once
Teredo
again,
Portal.
I will find that and I will put that in the description on YouTube as well.
Brilliant.
But I've just
Brilliant.
found the link here and I'll put that in the chat for the person who asked. That's wonderful.
Excellent.
Right, we're just past 8 o'clock. So
Yes.
thank you ever so much for that, Alison. I thoroughly enjoyed it. I hope everybody else
Good.
enjoyed it. I can imagine
That's good.
myself um, watching it several times, rewinding backwards and forwards, bringing back some specimens and double checking.
Mm.
And I think that's going to be really useful for people into the future because
Brilliant.
on our YouTube.
Brilliant. Yes, because I did have to move quite swiftly through, and there's a lot to take in. So I was really pleased it was going to be available afterwards for people to check back. And
Excellent.
thank you everyone for coming.
Yes, thank you. It was great to have 70 or so of you along. That's absolutely marvelous.
Yeah, fantastic.
Our next talk is next Tuesday on Equisetum. There'll be a link. I'll send an email tomorrow or the day after telling everybody that attended that this video is now on YouTube, but there will also be a link in there to the rest of our webinars. So if you're interested in Equisetum, and why wouldn't you be, then please come along to that next week. I think that'll be really interesting. And we'll also include various intriguing hybrids, I'm sure. So 2 exciting weeks on the trot. And with that, I will draw this to a close. Thank you, everybody, and I hope you have a wonderful rest of your night. Goodbye.
Bye.